Our study of Chapter 7 with the cell membrane and its function, as you would guess, we're going to break this discussion into two parts. And today's part will be dealing with the structure of the plasma membrane. The way the model of the membrane developed is a lot like the way that we've developed lots of different models for other things. When we started trying to make machines that could think, artificial intelligence, we started trying to do that, we all of a sudden discovered these are some of the ways that the really human brain works. So when we tried to build artificial membranes, that would do the same thing as the actual plasma membrane, we got a better insight, a better feel, for actually how real cell membranes work and function. Now, the way they tried to do it, they knew the cell membrane was made of phospholipids. So they tried to dissolve phospholipids and just see if they can get them to combine together. And the easiest way to do it was to dissolve them in a chemical called benzene. Okay, and that allowed the portions of the phospholipids to link together. And when you put those together, benzene will evaporate much faster. So when it evaporated, also they found out they had a little tiny layer, of phospholipids on the top of the water that was left. On closer examination, they found out that it was the heads of the phospholipids. The heads of the phospholipid are basically hydrophilic, so they are strongly attracted to water. So they stuck on the surface of the water, and all the little hydrophobic tails were left sticking up. And again, this all came about, these are called micelles, M-I-C-E-L-L-S, when they first started trying to develop these and make little tiny bubbles of phospholipids. That was where our first development came, but obviously these models left a lot to be desired. They don't function as a real cell membrane does. Now, our study of the cell membrane really took a quantum leap forward here when they actually started taking electron micrographs of the cell and actually started breaking the cell membrane down and studying its components. Through the chemical analysis, they found out that the phospholipids outnumber the proteins that made up the cell layer by a ratio of 2 to 1. And the first electron micrographs of the cell membrane showed, what looked like a protein sandwich. It looked like, for the early photographs here, it looked like there was two phospholipid layers on the outside and the inside, sandwiching a protein layer in the middle. Well, that didn't pan out on further investigations. Today, biologists accept what's called the fluid mosaic model of the plasma membrane. Fluid mosaic basically means that the membranes, while we say they're fluid, they actually behave like a fluid. For example, in this new model, there's very, very weak attractions, holding the membranes together. So if you look at this structure, this diagram we have right here, these phospholipids, and this again is a phospholipid. The round circular portion here is the head, which is hydrophilic. So it's going to be attracted to the water on the outside, and these phospholipid heads here are the heads are going to be attracted to the water on this side. Okay, so what the fluid mosaic model is saying is just that these phospholipids are capable of moving back and forth, and on occasion they can even flip-flop. So one from the inside can flip to the outside. So that means that the plasma membrane, instead of being stationary and unmoving, is constantly changing. And if you stop and think about it, when you've seen pictures of where they do cloning experiments, have you ever seen the times where they've pulled the nucleus of a cell out and inserted in some different genetic material? Why doesn't the cell just explode? Well, as the microneedle goes in, the phospholipids basically spread apart because they can move. So it makes that kind of surgery possible. Now, our last bullet down here, this one right here, where I noted that both the lipids and proteins can move, I should make note here that the proteins, they can move, but they don't always move. As you're going to see in the future, probably in the next slide here, some of those proteins are held in place by the cytoskeleton. Remember that part, those fibers that give the cell its shape. They give it shape because they will reach through there, stretch through the cell, and actually link together the different proteins. So proteins move, but not nearly as frequently as the phospholipids. Okay, now with the fluid mosaic model, everything moving back and forth. Okay, one of the things we want you to understand here is how these things can stay fluid. And the answer falls in the degree of saturation of the hydrocarbon chains. Okay? The more unsaturated hydrocarbons that you have, more unsaturated fatty acids in the tail portions. Again, this portion right down here. Here's the head. And these two stringy lines down here are the phospholipid tails made of fatty acids. The more unsaturated the chain is, the more these are actually kind of pushed apart. Okay? That's going to help keep the plasma membrane a liquid at low temperatures. Okay? As opposed to saturated fatty acid chains, they're going to make those molecules line up like little soldiers down here and keep the distance pretty uniform. All right? This arrangement right here that you see in like winter wheat, for example, helps the cells remain pliable in low temperatures. A cell that had this would be less pliable in low temperatures. Now, again, the one on the left is characteristic of some of the plants. How about animals? Well, animals, we have our own natural built-in antifreeze, which many people just call a really bad substance, cholesterol. Do need some cholesterol, help maintain the integrity of your cell membrane. What cholesterol does, if you look at my drawing over here, cholesterol kind of fits in between these phospholipids, breaking them up here so it spreads them out. This is why animal cells can remain pliable in pretty low temperatures, but they're not immune from freezing. Anybody that's been outside on really cold days, or you've seen people with frostbite, the cells do rupture under extremely low temperatures, and that is prevented to as large extent by cholesterol. This is probably a pretty good model right here. And as we look through this, remember, there's always a relationship between structure and function. Now, in the last section, we were looking at cell organelles. We said the cell membrane is kind of like a fence or some sort of barrier. Allows some substances in, keeping others out. Or vice versa, keeping some substances in and letting some out. It works both ways. We call that property selectively permeable. And cell membranes have several key components of their structure that make them selectively permeable. Now, some of the characteristics you need to make sure you're clear on before we look at how structure and function are interrelated is identifying some parts from this picture. We'll start with a phospholipid bilayer. Just to make sure when you're looking at a drawing like this, you're clear on where we're at. This part right here, these oval heads right here, these are the heads of the phospholipids, and these yellow portions right here are the lipid tails. So this right here, from here to here, there's two layers, phospholipid bilayer. Now, you also have in the cell membrane proteins. Now, the proteins are going to be either integral or peripheral. The difference is this. Integral proteins extend from one side of the cell, say for example the outside, all the way to the inside. Integral proteins are responsible for providing channels or tunnels that will allow certain substances to pass through. As we start looking more at the function of the cell membrane, you'll see the value of this. Because like we said in the previous bullet up there, membranes are selectively permeable. Well, how can they keep some things out and let others in? Well, one solution here is the integral proteins, where they're going to form a tunnel that's just the right size, just the right shape, just the right, chemical composition to allow that substance that's necessary to pass through either one way or the other. Peripheral proteins, they attach to the cell surface. They don't pass all the way through. Okay, sometimes the peripheral proteins are linked to integral proteins with parts of the cytoskeleton. The authors of this textbook try to show this down here. Integral protein here, and here's intermediate fibers with a cytoskeleton going through there, and they're trying to show that they're showing some linkages here, helping tie these proteins together and keep the cell shape consistent. Okay? Now, membrane structure for the same picture here, you've also got carbohydrates attached. Okay? Now the carbohydrates are right up here on this picture, and you're going to have a model here in a little bit here that I'm going to give you to work with. These are carbohydrate chains that branch off the cell. Like here's one, here's a couple more through here. Now the carbohydrates, as I put up here, cell-to-cell recognition. For example, if you have type A blood, okay, you have a certain carbohydrate that extends off your cell membrane. And just for convenience sake, just call it the A group. If you have that carbohydrate streaming off of your red blood cells, well, you've got type A blood. And we'll talk more about the inheritance of those traits as we go along, but transfusion rejections, transfusion problems, or transplant rejections, these are all critical here to make sure that these proteins are all alike. This is your body's way to recognize self from non-self. How does your body know that it's got somebody else's kidney in them, for example? Now, one last characteristic about these carbohydrates is they are technically oligosaccharides. Oligo is a prefix which means anything between 3 to 15. Now I know in our previously we talked about anything more than 2 we call a polysaccharide. As I looked ahead at the requirements here, I felt a need to identify oligosaccharides. Oligo, prefix which means anything between 3 to 15.